Thank you for asking me. I don't really mean that. That's a lie, too. Anyway, I'm here, and I'll do my best. What I'm going to try and do is put this drug discovery business in some kind of perspective. I think searching for new medicines is the only truly unique thing about human beings. Some of the earliest human records, such as the Egyptian Ebers Papyrus, written over 3,500 years ago, dealt with medication. Everything in nature seems to have been drummed into the service of seeking relief from distress. To get relief, they ate herbs, woods, roots, juices, minerals, and animal appendages. Our ancestors must have been avid experimentalists. The results of these experiments were passed down by word of mouth, folklore medicine. A few years ago, I had the opportunity to visit a museum in Heidelberg in Germany. The museum authentically recreated the pharmacies, the official repositories of folklore medicine, which operated in Germany from the 14th to the 18th centuries. These pharmacies administered the official pharmacopoeias, that is, the currently accepted list of remedies and nostrums of the day. The museum shows how the pharmacopoeias kept changing. As products fell into disfavor, new products were recommended to fill the gaps. We now know that many diseases will ameliorate for a time, let me emphasize that, for a time, by giving biologically inert substances, placebos. So are these pharmacopoeias therapeutically illusory? Probably most of the remedies were worthless, but folklore medicine had its successes. Some of them are still used in modern medicine. Roots of the belladonna plant are now known to be a source of atropine, leaves of the foxglove, source of digitalis, seeds of the poppy giving us morphine, cinchona bark giving us quinine, and so on. I could list many more. Now, how people originally found out about the therapeutic properties of plants is truly a mystery. Although the discovery of the properties of the bark of the willow tree is supposed to have had some kind of logical basis. Arthritis and certain fevers became linked in the medical mind with living in damp conditions. It's not normal for people to live in damp conditions. However, willow trees thrive on damp conditions, therefore they must have a natural mechanism to protect themselves from the damaging effects of dampness. The ground-up bark of the willow tree was found to relieve the pain of arthritis, an effect now known to be produced by salicylic acid. So this empirical approach to therapeutics did not give way to a scientific approach until the middle of the 19th century. To be precise, the year was 1856. Does that work? Will it work? Oh, all right. All right. This is William Perkin. When William Perkin was 18 years old, he was doing amateur chemistry experiments on the top floor of his parents' home in East London. At that time, London was heated and lit by gas derived from the distillation of coal, a process that produced heaps of useless byproducts, including coal tar. Hoffman, Perkin's tutor, had shown how naphtha extracted from coal tar could be converted to the artificial alkaloid known as naphthalidine. Perkin was trying to convert this artificial alkaloid into the natural alkaloid quinine. Quinine was in short supply and very expensive, but vitally important to prevent the malaria that was rampant in Europe and the USA, as well as in the regions of Africa and South America that the European imperialists were trying to conquer. His experiments failed, but he did produce a magnificent new color, mauve. Mauve led to a whole new industry for synthesizing artificial colors, aniline dyes, and this led on to the development of explosives, perfumes, photography, and modern medicines. But the contribution to medicines came from Paul Ehrlich. By the time Ehrlich went to university, he had developed a passion for microscopy and for staining tissues with aniline dyes. Although he was not the first to use aniline dyes to stain tissues, he was the first to develop a theory on the staining process. While he was an undergraduate, he discovered mast cells, not their function, of course, that would have to wait for nearly a century. When he graduated in medicine, he was 24, and in his graduation thesis on the theory and practice of histological staining was remarkable. From contemplating how foreign chemicals came to stain cellular components so selectively, he mapped out a lifetime's research career. His early ideas about chemical affinity would lead him onto his work with von Behring on antitoxins. His work on antitoxins led him to the development of his side chain theory, his ideas about receptors, and his concept of magic bullets. When he tried to explain his ideas to Robert Koch of Koch's postulates, he ended up drawing with chalk on the floor figures like these. He imagined that cells sprouted finger-like processes, which he called haptophores, that could stoichiometrically bind toxin molecules with their adsorbed toxophore groups attached. At the top left, there is his idea of a receptor of the first order, and the other show his ideas of the second and third order, which involve binding of nutritive molecules and complement molecules. Contemporary immunologists would use a different vocabulary and images, but the underlying concepts would not, I guess, be very different. His discovery of vital staining with methylene blue led him to develop chemotherapy as a concept-driven goal. He aimed to find dyestuffs, arsenicals, that would selectively kill parasites with minimal damage to the host, which he measured as the therapeutic index. His last discovery was that dioxydiaminoarsenobenzene was highly effective against experimental syphilis. It was the 606th compound that he tested. It's now known as salvarsan. So was Ehrlich the first to use random combinatorial chemistry? And the answer is no. He first showed, I'm sorry, this is a rotten slide, you can't possibly see that, but if you could have seen it, there's two structures on there. One is atoxyl, which was known at that time as a highly toxic parasiticide, produced blindness. And the structure which had been allotted to it, Ehrlich showed was wrong. It wasn't a non-reactive analyte, but a reactive aminophenyl arsenic acid. Starting from this, he systematically changed its structure using chemical ideas he had developed in a lifetime of dyestuffs chemistry. Ehrlich was truly the father of medicinal chemistry. When Domack joined IG Farben Industry, a manufacturer of azo dyes, as research director in 1927, he was inspired by Ehrlich to test their dyes against hemolytic streptococcal infections in mice. In this way, he discovered the bacteriostatic effects of Prontosil Red, the forerunner of sulfonamides, and a chemotherapeutic revolution. He even treated and cured his own daughter. Finally, the most prolific drug discovery of all time, Paul Janssen, also used Ehrlich's systematic, iterative approach. Just as Ehrlich's head was full from years of experience of an array of chemical groups that he referred to as chromophores, so Dr. Paul, as his researchers knew him, had from years of experience his head full of pharmacophores. I've watched Paul many times at meetings. When he gets bored, he draws new chemical structures on his program. He began his research by starting from pethidine, partly because he knew that it was a fairly simple molecule, and partly because he knew that piperidine chemistry was easy. I have a slide of this, which you won't be able to see either, but I'll just put it up. Pethidine was a relatively new drug at the time, and it was being used as a substitute for morphine. But it had originally been introduced into medicine as an antispasmodic. So Paul proceeded systematically to modify its structure and succeeded in separating the pharmacological properties of analgesia as fentanyl and its antispasmodic antidiarrheal properties as loperamide, both still highly successful drugs today. From then on, chemically speaking, one thing led to another. And between 1956 and 1984, he invented and brought to market 35 new drugs. No one has ever achieved such success. Thus, these three great men had in common, one, that they started from molecules that they already knew had selective affinities for tissue components. 2, that new compounds were tested in whole animals, or in Janssen's case, sometimes using isolated tissue bioassays as well. And 3, that the pharmacological results of these tests inspired the synthesis of new molecules based on informed ideas about structure-activity relationships. They all used the kind of iterative, evolutionary, selectionist approach that Darwin had recognized as the basis of successful biological speciation. It is axiomatic that all new drug research programs have to start somewhere with a chemical program. Ehrlich and Domach started from dyestuffs, Janssen started from pethidine, and in 1958 I started from the knowledge of the tissue selectivity of isoprenaline and Alquist's idea of receptor subtypes. However, before giving you, as an example, a very brief history of the beta blocking story, I want to go off on a short loop about receptors as a basis for new drug discovery. What I'm really doing is taking the opportunity to get a beef off my chest, and it's really got nothing at all to do with the drugs, but I'm here, and you're there, and I'm going to get it off my chest. Pharmacologists talk about receptors as though they have proprietorial rights to them. But receptors are broadly defined in my dictionary as any devices that receive information, signals, etc. And as such, this concept is widely used in biology. So physiologists refer to sensory receptors, chemoreceptors, and baroreceptors, while microbiologists talk about viral receptors. And ever since Langley, since about 1876, in fact, pharmacologists have talked about drug receptors. So receptor only has meaning when it carries a prefix. The problem with Langley's prefix of drug receptors was that it did not carry any physiological implication. For years I have proposed, this is the beat bit coming, that pharmacologists should do the same and use the prefix hormone to the receptors that interest them. Hormone, a word derived from the Greek word for messenger, was invented by Bayliss and Starling in 1902 as a metaphor for the physiological role of secretin, a messenger molecule that happened to be carried from duodenal mucosa to the pancreas by the circulation. Since then, unfortunately, chauvinistic pharmacologists have decreed that if a messenger molecule is not circulated, it is not a hormone. What a pity. Dear old Starling did not know about the chemistry of neurotransmission. So we now have a mess of neurotransmitters, paracrines, autocrines, autocoids, whatever they are. However, why not let us classify them all as hormones, messenger molecules, and the conjugate receptors as hormone receptors, as the iconic receptorologist A.J. Clark proposed ages ago. During the last 100 years or so, the evolution of our ideas about hormones and the receptors has been exponential. However, even I find it hard to believe that a mere 40-odd years ago, physiologists did not recognize hormone receptors, and physicians never talked about them. Receptors? What are they? Endocrinologists, of course, talked endlessly about hormones, but never felt the need to talk about their cellular receptors. The only people who talked about receptors were a small group of mathematically minded pharmacologists who tried to understand the quantitative relations between dose and response by applying the law of mass action. For their equations, they needed the drug molecules to interact stoichiometrically with unspecified tissue molecules. They liked their idea, but I remember talking to them in the 50s, and they were certainly embarrassed about the fact that of the molecular vagueness of the ideas which they were using. So in my time, I have seen the concept of a hormone receptor change from one, after Langley, being a chemical concept, the receptive substances and tissues that drugs react with according to some law of combination. After A.B. Hill, a mathematical operator, the hypothetical chemical component that drugs interact with to be used as a concentration parameter in algebraic equations. After Alquist, an explanatory model, Fersby used him to explain the selective tissue effects of isoprenaline. 4. Consequently, we learned to use this concept as a basis for seeking selective drug actions. Then we learned that receptors triggered the production of specific intracellular second messenger molecules. And finally, as you've been hearing from Bob, receptors have been extracted as unique chemical entities and then shown to be specific gene products that can be inserted into the cell membranes of naive cells. Overall, it's a quite breathtaking exercise that has taken about 50 years. And venture capitalists and politicians are looking for similar progress today in 2 to 3 years. And the question in my mind is, when will they learn that they are looking in vain? Of course, the beauty of using hormone receptors as targets is the lead molecule has itself got the highly selective drug-like qualities that we want. So back to my theme. An adequate description of a hormone receptor system requires evidence of hormonal efficacy. Hormonal efficacy can only be measured with confidence in intact tissues. Efficacy changes in intact tissues can only be interpreted by molar models. And so it is that the early ideas of receptors as mathematical operators have turned out to be tremendously useful experimentally. Ideas about the concept and tests for competitive antagonism were first proposed by A.J. Clark from the parallel displacement of acetylcholine dose response curves on the isolated frog heart. And from this, he developed the idea of the dose ratio. This concept of measuring drug antagonism by the degree of parallel displacement of dose response curves was further developed first by Gadam and then refined by my guru, Heinz Schilt. The eponymous Schilt plot that relates the log dose ratio -1 to the antagonist concentration lies at the heart of operational analytical pharmacology. Model displays of, Some time ago, Nigel Shankly replotted Clark's data, which I just showed you, in Schilt space. So you now see the straight line with which we're more familiar, with the slope of 1, and is the model display of simple competitive antagonism between acetylcholine and atropine. You already, if you look at that, can see the problem, because atropine there has a PKB of just over 7, whereas a mammalian tissue is just a PKB of over 9, a difference that cannot be accounted for thermodynamically by making the frog measurements at room temperature. So I wonder what that means. And the truth is, we know so little about comparative pharmacology. Anyway, I was taught this simple model of and test for competitive antagonism by James Raventos, A.J. Clark's pupil, whose lab I was lucky enough to share when I joined ICI in 1958. Raventos also taught me about how Arians and Stephenson had independently discovered that hormones behaved as though they had two properties: the hormone binding to its receptor, the property of affinity, and then the property of efficacy, the ability to activate the receptor. The exciting discovery was that affinity and efficacy were separable properties. For me, that meant that there should be a way of converting a hormone into its own competitive antagonist. So that was the problem for me, how to find a competitive antagonist for adrenaline at beta adrenoceptors. But why did I want to do this? The beta blocking story grew in my mind with clinical, therapeutic, physiological, and pharmacological elements. Clinically, angina pectoris was known to be precipitated by anxiety and emotion as well as exercise. At one time, an injection of adrenaline was used as a test for angina, long since abandoned because the test passed too near the cemetery. Partial thyroidectomy was found to relieve severe angina for a time, until induced hypercholesterolemia aggravated the underlying atheroma. The connecting link between these three seemed to be tachycardia. Therapeutically, nitroglycerin could quickly relieve an attack of angina, associated as it was with a facial flush, and it was assumed that the coronary arteries were dilating as well. But new selective coronary vasodilators, such as dipyridamole, were developed by the pharmaceutical industry and were found to be clinically ineffective. Biologically, Smith and Lawson, working in my lab in Glasgow, found that hyperbaric oxygen at 2 atmospheres pressure reduced the incidence of ventricular fibrillation in dogs after coronary artery occlusion, although the increased oxygen capacity was not more than about 20%. So would an equally small decrease in oxygen demand be just as effective? That was the question. The main determinant of cardiac oxygen consumption is heart rate, and the main determinant of heart rate is cardiac sympathetic nerve activity. So these clinical, therapeutic, and physiological features of hearts coping with coronary artery disease pointed to the potential advantages of annulling the actions of noradrenaline and adrenaline on the heart. Pharmacologically, anti-adrenaline drugs were a well-recognized class in 1958. All of them showed a pattern of actions similar to those seen by Dale in 1906 with ergot alkaloids. That is, they would reverse the oppressor effects of adrenaline, but did not block the induced tachycardia. In 1940, Conzett had shown that the isopropyl derivative of noradrenaline, isoprenaline, or isoproterenol, perhaps I should say on this side of the pond, stimulated the only actions, such as tachycardia, that the anti-adrenalines couldn't block. It had been introduced into medicine as a bronchodilator. And Ray Alquist was having difficulty trying to explain the actions of isoprenaline on the basis of Cannon and Rosenbluth's hypothesis of sympathin E and sympathin I. This is Ray at about the time he was doing the work in Iowa. So he proposed that the actions of adrenaline were mediated by two classes of receptor that he referred to as alpha and beta. No one had ever used this receptor idea before to explain the selectivity of drug actions. The anti-adrenaline drugs of the day were therefore alpha receptor antagonists, and isoprenaline was a selective beta receptor stimulant. So I went to ICI in 1958 to find a beta adrenoceptor antagonist. John Stevenson was the medicinal chemist allotted to work with me, and I was given one laboratory technician, David Dunlop. So that was us. Are there smaller drug discovery groups anywhere today? The structure of isoprenaline was our only clue. Apparently, higher orders of N substitution lost affinity for alpha receptors. So then we thought perhaps substitution with a different but larger group on the nitrogen might produce a selective antagonist. So what we were looking for were substituted dibenzyl ethylamines as interesting drug targets. Well, we used a Langendorf preparation, the ordinary classroom experiment to start with. We made compounds, we tested them, and we were just getting nowhere when we read about Powell and Slater's invention of dichloroisoproterenol. They were hoping to make a long-lasting bronchodilator, but in fact produced an isoprenaline antagonist. However, on our bioassay, DCI was a full agonist like isoprenaline. I'd also developed the technique for simultaneously recording blood pressure and heart rate in analog form in anesthetized cats. When DCI was given by slow intravenous infusion, it produced a huge increase in heart rate, as you can see. So we rejected DCI as a lead, but meanwhile, I developed a new assay, the electrically stimulated guinea pig pupillary muscle of the left ventricle. Retesting DCI, we found now that, as you'll see that on the right, it was not an agonist, but in fact blocked the stimulant effects of adrenaline. And on the left side, you can see the agonist effects on the right atrium. So this was my introduction to the tissue dependence of partial agonists. John immediately got the idea of making the naphthyl analog of isoprenaline, knowing that the naphthyl ring would have similar steric but different electronics from the dichloroisoproterenol. So we took pronetholol into man for what we now call proof of principle studies. Here is the first of Dornhorst's volunteers, where blood pressure, respiration, and forearm blood flow by plethysmography were measured during an infusion of isoproterenol. As the heart rate increased, respiration deepened and blood flow increased. And the volunteer told me afterwards that he thought he was going to die. There's a tremendous, apparently, when your heart goes racing away and you're lying on the bed, it's just awful. However, the isoprenoline repeated along with prenetholol, and all the changes were blocked, including all this fear of doom. Now here is the first patient that we gave the drug to. I think my gadget is falling apart here. Do you mind if I just do some surgery on it? I think I'm there. Just to see. Yes, okay. So here's the first patient showing the onset of angina after 5 minutes of exercise. As the exercises, the rate increases, and then when it gets up over 100, he gets his attack of pain and has to stop. And then when he repeats this after prenetholol, the patient can now do more work and exercise for longer before the onset of pain. Now, although I've never thought that prenetholol was good enough to be a drug, the discovery of thymic lymphosarcomas in mice, there were 2 of them in fact, finished the drug off. So the backup compound propranolol was not long in coming, and then we showed that both of them could produce parallel displacements of dose response curves. These are the spontaneously beating guinea pig right atrial preparation, and the Schultz plots are shown down below. Now, just now, I'm going to just show you Nicola here preparing a heart. No, it's not that, the wrong one, damn it. I meant to show you, I wanted you to look at something when I'm going to read for a while. And I was going to put up a pretty gal for you to look at. I thought that would be more interesting. But you've put them the wrong way around. That was going to come last. Anyway. Right. So now I used the same procedure to invent the H2 receptor antagonists. At the Wellcome Foundation in 1976, I started a similar program around 5-hydroxytryptamine, one that Graham Martin eventually brought to fruition with the invention of zolmitriptan, a 5-HT1B agonist. More recently, using the same principles, my group has invented antagonists for CCK1 and CCK2 receptors, and also for histamine H3 receptors. Now, in none of these cases was the molecular biology of the receptors known before we started. Now, this approach to the invention of selective hormone receptor antagonists has been used successfully by many groups. Indeed, the invention of many different hormone-specific competitive antagonists has been one of the great success stories of the pharmaceutical industry. So this is the background to my belief that it's possible to generalize a strategy for drug research. So a drug research strategy is the set of principles which help you to decide what to do in the lab with the clinic in mind. Based on my own experience and reading about other people's experiences, I can tell you what mine are. What we want from a new drug is a pattern of selective effects, which, if they occurred in man, might be imagined to have therapeutic merit. The first principle, then, is that I must have evidence before I start that the desired selectivity is capable of being expressed by a drug. There may be empirical evidence that other drugs are already known to possess that selectivity, and then there is no issue to settle. However, when the desired selectivity is new, then unless there is evidence of the biochemical differentiation which will allow the selectivity to be expressed, then the project is likely to be based on mere wishful thinking and probably doomed from the start. Wishful thinking in drug research is enemy number one. The second principle is that there must be an explicit chemical starting place. Medicinal chemists must have a molecular template with the command, begin here. The chemical template may be an already known drug, or it may be a natural, physiologically important molecule, or even today, an HTS-generated lead. But they have to start somewhere. The third principle is that there has to be a biological measurement of bioassay, which can measure the specific property imagined to underline the intended selectivity. In my experience, a functional bioassay is always preferable to a surrogate marker. The fourth principle is that before I start, I have to be able to imagine how I will be able to show in man that the drug has the same basic actions as I would have hopefully shown it in the laboratory. The fifth principle is that I have to have some disease in mind, in which it will be logical to evaluate the new drug's properties. The sixth principle is that I or one of my colleagues must feel passionate about the project. Drug research programs can go through long periods, as much as 2 to 4 years or more, when little progress seems to be happening. Then passionate conviction is what keeps you going. Finally, the seventh principle is I don't start a project unless I know that there are funds to complete it. Now, notice that I make a calculation about costs, but not about markets or sales. I like to make my research strategy quite explicit and encourage my research group to wrestle with its practical implications. At its best, I think this is how the inventive process works. Once the chemical structure of a hormone, say, is known, the medicinal chemist can, in his imagination, walk around the molecule and try to guess what are the chemical features that might make it efficacious. An initial guess can be evaluated by making an appropriate analog or derivative of the hormone. The new compound has to be evaluated by a bioassay. As I've explained, a bioassay is a piece of tissue which responds characteristically to the hormone. The new compound can be tested alone. Is it still efficacious? And then in combination with the hormone, does it antagonize the hormone? At this stage, negative answers are as informative as positive ones. Either way, the results suggest new chemical questions, new chemical compounds to be made and tested. This basic iterative loop, synthesize, test, synthesize, has been shown by everybody who's tried it to work reliably. Compounds with a partial loss of efficacy are followed by weak antagonists, leading eventually to very potent antagonists. The only unknown is how long the process will take. In my case, always about 4 times longer than I had hoped or expected. In many ways, this initial stage of drug research is the most intellectually and psychologically demanding. My approach is this: if the grounds for feeling passionate about the project at the outset were sound, then total commitment is called for as long as is necessary. John Locke, the 17th century philosopher and scientist, talked about making his discoveries by steadily intending his mind in a given direction. Leonardo da Vinci talked about the need for obstinate rigor and creativity. In some ways, the early stages of drug research are more about character than intellect. So far, as you can guess, I've been describing the search for new drugs in the pharmaceutical industry as I have known it for 35 years. I've also been describing the principles by which I now run a very small 25-people drug research organization within the environment of King's College London. However, within the last few years, an extraordinary revolution has taken place within industrial pharmaceutical research companies, as Bob Ruffalo has been explaining to you. The R&D revolution has occurred at a time when the major pharmaceutical companies have never before enjoyed such prosperity. The upside of the market is that every major pharmaceutical company reported double-digit profits last year. The pharmaceutical market is unique. The desire for medicines is preemptive. People, even very poor people, spend money on drugs in preference even to food. The desire for drugs is inelastic. There are no trade cycles, no slumps in demand. The desire for drugs as a commodity is uniquely international because to a large extent, we as human beings around the world suffer from similar ailments. The downside of the market is that the monopoly phase, as you've heard earlier, of patent-protected marketing is being progressively eroded as obligatory development times have increased. At the same time, there is huge pressure from governments and health maintenance organizations to reduce the prices even of monopoly drugs. The business end of the pharmaceutical industry is consequently becoming impatient. More products are needed. If they cannot be achieved by mergers and takeovers, then the in-house R&D productivity has to increase. And so it happens that these commercial constrictions have been occurring at a time as technological revolutions have been occurring in biology and chemistry. You just heard about all that. It's been driven, as you've heard, by extraordinary advances in genetics and molecular biology. As a lead generator, CombiChem plus HTS undoubtedly works, as you've heard. Perhaps this is not surprising. Complexity specialists, such as Gerald Kaufman, argued that life began as an exercise in combinatorial chemistry. Primitive bacteria-like and tube-like forms seem to have been around for over 2.5 billion years before the Cambrian explosion started structural evolution 500 million years ago. Maybe natural selection had first to operate at the level of combinatorial chemistry, chemical evolution, to develop populations of molecules that were comfortable with each other, before structural evolution could take off. My reading of the history of drug inventions suggests that the most selective drugs, with the widest therapeutic ratio, have come when the initial lead was a native physiological molecule. Perhaps drugs that are crafted around a natural template retain some of the parental selectivity. So my caveat to the CombiChem, my first caveat to the CombiChem plus HTS strategy is that leads may not all have the same quality. However, my second concern about the current thrust of drug research is that it is rooted on targeting components rather than systems. The importance of working with systems was discovered by Fourscott nearly 20 years ago, work that led to the extraordinary discovery of nitric oxide as a universal messenger. Now, this is such a good story, I'm just going to retell it because I like telling it. When acetylcholine is infused intraarterially, the blood vessels dilate. Fourscott studied the effects of drugs on arterial muscle. Oh, there's Nikki. Oh, I should have kept going. There we are, here's Fourscott. I'll bring her back at the end if you'd like to. At one time, Fourscott studied the effects of drugs on arterial muscle, like I'm showing you in the slide, in isolated organ baths. Now, at one time, the only way to measure the effects of drugs on arterial muscle was by measuring changes in its length. To get measurable shortening, the blood vessel was cut into a long spiral strip. In this preparation, Fourscott found that acetylcholine had no relaxant effects. Indeed, the muscle usually contracted, bottom right-hand corner. Imagine his surprise. No, I'm sorry, I missed a sentence up there. When instruments for measuring tension became available, isometric measurements could now be made on rings of arterial muscle. So you can imagine his surprise when he showed that the endothelium, that acetylcholine now had the expected relaxant effects on arterial muscle. Fourscott showed that the endothelium was intact in the muscle ring, but destroyed in the muscle spiral. He showed that acetylcholine relaxes arterial muscle indirectly by stimulating the endothelium to secrete a relaxing factor, which diffused into the adjacent muscle layer. The seminal discovery was only possible when the muscle and endothelium was combined in a system. We now know that the relaxing factor is a gas, nitric oxide, which escapes when endothelial cells are cultured in vitro. So this astonishing discovery could not have been made by the powerful component-directed techniques of molecular and cellular biology. Whatever the explanation, it is becoming clear that even in these simple in vitro systems, we can see complex pharmacological behavior that would be missed by studying drug actions at the chemical level. Finally, I want to make a more general and speculative point about the contrast between systems and components. As far as I can see, the new drugs that the pharmaceutical industry is seeking to treat disorders such as asthma, dementia, and cancer are expected to be similar to the ones that they have already invented to reduce high blood pressure, heal stomach ulcers, and relieve pain. I am concerned that this expectation may not be fulfilled. Physiology is about how cells use chemicals to talk to each other. Sometimes the message has the shape of a command, such as contract or secrete. Thus, noradrenaline is the final messenger to the pacemaker of the heart in emergency situations. So a drug that blocks the effect of adrenaline on the heart effectively controls cardiac stress responses. Note that the heart must react to stress reliably, on cue, but if it beats faster inappropriately, nothing very bad happens. The process of heart rate changes is inherently reversible. Some of our most useful drugs act by interfering with chemical commandments of this type. However, there's another kind of physiological system that must always be activated reliably on cue, but which, if activated inappropriately, can have damaging, even lethal effects. Examples of these systems are commitment of bone marrow stem cells, activation of killer lymphocytes, cell division, growing new blood capillaries, and so on. Now, the feature of these is that once initiated, these are inherently irreversible processes. So how are these physiological processes controlled such that they can be activated on cue, but never inappropriately? The striking feature of these irreversible processes is that many chemical messengers are involved, each having a different cellular origin. They are often described as cascades, but they are unlikely to operate in sequence as implied by that word. Another feature of these messengers is that they can often be shown to potentiate each other. So I imagine a process that I call convergent control. I imagine that an effective stimulus might involve the cooperative interaction of more than one agent involving addition or amplification of individually subliminal stimuli. I imagine a growth factor giving a stem cell, not a command, but a piece of advice, such as, other things being equal, you should start dividing. The other equal things are other chemical messengers, which have to impinge on the cell at the same time to achieve its activation. Unlike the command-control action of adrenaline-like molecules, such an advised consent arrangement leads to information-rich management. Physiological control by chemical convergence entails the possibility of redundancy. Biotechnology has been hugely successful at blocking various molecules that are overproduced in the immune system in septic shock. In every case, laboratory success has ended up in clinical failure. At some point, we must ask whether the model or our way of thinking is wrong. I believe that the way forward will not lie in the discovery of more and more components, but in improving our understanding of how components are organized into systems. But seriously, how can we study complex systems in the lab? Now, to illustrate this, I have a slide that I use quite a bit. It's simply a list. It starts at the top with populations, clinical trials, if you like, and then we have isolated organs, pieces of tissue, with architecture and text, spanning organ bus, cells and tissue culture modules, all the way down the list, you can see it. I used to say that this was my reductionism slide, if I viewed it from the top down, or my hierarchy slide if I viewed it from the bottom up. However, with Mandelbrot's approval, I now say that this is my fractal slide, because I've come to realize that reductionism in biology does not achieve the aim of simplification. Reductionism in biology merely replaces one type of complexity by a different kind of complexity. No one level is more reliably informative than any other. So I strongly believe that pharmacology needs to be studied at all levels, the choice of level being dictated by the nature of the question being asked. The choice of level for studying the pharmacology of complex systems is the intact tissue bioassay. The attractiveness of these bioassays is that they can be driven chemically and physically in as many ways as our imaginations can conceive, and yet still remain, potentially at least, mathematically tractable and analyzable. So I believe that intact tissue bioassays have an important future. Searching for new medicines, I have to tell you, is an exciting and rewarding activity with great opportunities for the future. I wish I could start all over again, and I would study chemotaxis, how cells move to touch other ones driven by chemical communication. There will be new drugs in there somewhere. Thank you.
Jim is willing to answer a few questions, so if you have a question, can you please stand up and speak quite strongly?
See anybody?
No, I can't see anybody out there either.
Speechless.
So James, thank you. That was just wonderful. You began with a charming description of something called the doctrine of signatures. It seems to be invented the same way, suggesting that we have to go to what is actually working to find drugs. Is that the message?
That is the message. The message is that I think Bob Ruffalo gave us the same message. I may be suggesting a little more explicitly where we should be going. But biologically, we have to forget about components. We've got to think systems. And it's a whole new field we have to learn. But I think, I find it very, very sad the way we're so concentrating now at the molecular end. Can you hear me? At the molecular end of things, that we're not any longer studying the integrated physiological thing. Back to physiology, I say.
Are there any more questions? That being the case, there being no more questions, I think the score is reductionist 0, functionalist 2. I would just like to remind you that we have a reception at 6 o'clock. That does include food, wine, and poster viewing. And that is all located in the big room next door. So you may all have a break now and be back at 6. But I would just ask the WPS judges to meet me outside in the registration place. Thank you very much.